If you could change the world, would you? Our world is constantly changing, largely because of human innovation, whether that's good or bad. But what if we could go even further by fundamentally changing living creatures down to the base pairs of their DNA? If you could revolutionize global agriculture, make food more nutritious, and reduce hunger, despite any unforeseen environmental risks, would you do it? Before deciding, it's important to understand the basics of the CRISPR-Cas9 system. CRISPR stands for clusters of regularly interspaced short palindromic repeats. It may be a tongue twister, but basically it's just a collection of genetic information. Cas9 stands for CRISPR-associated protein number 9, and it is the protein that reads genetic information. So let's get into the process of how CRISPR works. Viruses attack by attaching to the outer wall of bacteria. The virus then injects genetic material inside the bacterium with the goal of mutating the cell to create even more viral DNA. Most cells do not survive the attack. The ones that do survive store a piece of the viral DNA in a database known as CRISPR. When the bacterium is attacked again by the virus, the bacterium makes an RNA copy from the CRISPR archive. The Cas9 then finds the RNA strand produced and searches through the bacterium's genetic material. Once viral DNA is found, Cas9 acts exactly like a pair of scissors. It cuts out the viral DNA, making it harmless. It does so precisely down to the base pair. It's surgical precision at a microbial level. The true discovery, however, is that the CRISPR-Cas9 system is programmable, meaning that scientists can act as CRISPR and give a Cas9 protein, a copy of DNA they have selected to target. This not only works on microbial cells, but plant and animal cells as well. Pros. The greatest application of CRISPR in the agricultural community is its potential to increase food security, because climate change has the potential to impact global agricultural production in every aspect. Researchers at the University of Bonn have calculated that global food production could fall as much as 20% by 2050. Karl-Heinz Kogel at the Justus Liebig University in Germany is working on making wheat and other crops resistant to disease. He has done this by knocking out genes that are responsible for undesirable traits. This alters the wheat's genome and creates a variety of wheat which is resistant to mildew. This is important because as the earth warms, fungal infections are anticipated to spread further north. Adapting crops to a new climate condition through natural selective breeding can take up to a decade. With CRISPR, it can be done in weeks. Also, not only can we make crops resistant to disease, we can increase their yields. Zachary Lipman and researchers from the Cold Springs Harbor Laboratory have altered tomato plants in 3 ways to increase production. They increase fruit size, increase branching in the plant, and alter the shape of the plant. Cold Springs Harbor Laboratory researchers are also currently working with tomatoes to make tomatoes hardier in colder temperatures and potentially introduce tomato farming in Canada. Alterations can create new market opportunities globally. Canada could have a domestic supply of tomatoes to feed their demand. This could lower costs associated with produce imports and provide jobs within the industry. CRISPR makes it a possibility to introduce crops to other parts of the world that could not naturally experience them. New domestic markets could be established, developing economies that are primarily agrarian would thrive if crop variety increased within their nation. Using CRISPR, we can grow more in more places than ever before. Also, we aren't limited to plants. There are livestock applications as well. Livestock pig farmers are faced with the challenge of how big their pigs should be. If they're too big, it's too expensive to raise, and if they're too small, the pig will die from hypothermia. Farmers must find the right size because pigs are not efficient at regulating body fat. They lack a gene most mammals have for body temperature regulation. By using CRISPR to insert a gene into the pig genome, it is possible to make them efficient at burning fat and stay warm. Now pig farmers can earn profit from low-fat bacon, and we can even modify pigs so their organs are fit for human transplant. And there are still other livestock applications as well. A gene drive makes it possible to have only male or female offspring. It is possible to pick one and only one continuously. This can be extremely beneficial to cattle farmers. By being able to produce all male cows, the possibilities are endless. Oranges could be gene edited to grow in Ireland, bananas could come to America, low-fat bacon, and ground beef could be $1 a pound. but what are the risks? Cons. CRISPR technology has the ability to change the world, but does that mean we should let it happen? Like every new concept or technology, skepticism is always present. One of the major downsides to this new technology is the stigma that is associated with gene-edited organisms. A stigma currently exists with genetically modified organisms, and despite scientific reports. In order to isolate the stigmas associated with GMOs from CRISPR technology, the public needs to be informed of those key differences. With friction already present amongst consumers and producers, it could be extremely challenging to gain public support for implementation, especially since it has the ability to annihilate entire species. As mentioned with Terminator bulls, CRISPR can manipulate the sex of an organism's offspring. Gene drives are being applied to mosquito lab populations in an attempt to find solutions to the malaria epidemic. If not careful, ecosystems could be disrupted by species extinction. All of these concerns fuel controversy. One major issue came to light when scientists were testing out CRISPR on mice. They attempted to edit the genome of the mice, but as a result, there were mutations all throughout the genome, mutations that were an unintended consequence of the genome editing. It raises the question, if the genetic mutations occur in the mice, what is going to happen to the CRISPR food we will soon be buying at the grocery stores? The study found that issues with CRISPR was not completely reliable. Their sample size was relatively small and the data was recorded in a relatively short period of time. These are not characteristics of a data set that can serve as a benchmark in determining whether or not CRISPR is safe. Furthermore, the mice that were genetically modified using CRISPR were supposed to be genetically identical to a third mouse that was the control group. This was not the case and skewed the results of their testing even more. Policy. While the FDA during the Obama administration in 2017 said it intended to treat CRISPR-edited animals as if they were new drugs, requiring elaborate and costly safety studies, the matter is not so simple. Other parties believe there should be little to no government intervention on genetically edited organisms. Many companies lobbied to the Trump administration in favor of minimizing these policies, and Trump indeed promised to streamline regulations that have blocked cutting-edge biotechnology, setting free our farmers to innovate, thrive, and to grow. While this sounds wonderful for farmers, there is a debate as to whether it is safe to blindly embrace this new technology that has the potential to change the way we eat forever without taking cautious, preparatory measures in the beginning stages. As of now, however, according to Secretary Sonny Perdue, the USDA plans on freely allowing the use of genetically edited organisms so long as the result could have eventually been naturally bred, and it does not involve or create plant pests. Essentially, you cannot create an entirely new plant or make broccoli taste like bacon, but you can modify plants to be more resilient to disease and drought or increase their nutritional value. Perdue emphasized that although the USDA encourages the use of this biotechnology for its potential to increase food security and nutrition among Americans, it will not be free from regulation or research.
Given the arguments on both sides of this issue, we felt that a good intermediary policy would be the implementation of some sort of CRISPR certification, at least within the initial 5 years of its use. Through this policy, companies who wish to use genetically engineered organisms would have to pay the USDA to examine their practices and approve the product for safe sale. After being permitted to sell the product, the company would have to put a label on it saying these GEOs have been approved for safe consumption by the USDA, or something of that nature, to promote consumer trust. Companies using GEOs within the next 5 years would have to go through this process in order to sell their products legally. These steps would create a foundation for future use of genetically engineered organisms by stimulating research and consumer trust.